All right, hi, I'm Alex. I graduated from Geneseo in 2017. I was a member of the biology department. I was in actually Dr. Hutchison's lab. So now I'm a fourth year graduate student at UB in Sat Sinha's lab. And today I'm going to be talking to you guys about the novel mediators of the p63-driven oncogenic activity in squamous cell carcinoma. So my lab uses head and neck squamous cell carcinoma as a model of squamous cancers. So to give you guys some background on what HNSCC is, these are epithelial malignancies that arise in the upper air digestive tract, including the paranasal sinuses, the nasal cavity, the oral cavity, the pharynx, and the larynx. These diseases are unfortunately associated with poor prognosis and poor overall survival, and really no changes in disease outcome or disease treatment have been made in the past 3 decades. So there's a huge effort to actually molecularly characterize these diseases to hopefully find drivers of HNSCC that could lead hopefully to targetable downstream genes. So systemic sequencing studies have actually indicated that p53 family members p53, which we know is an important tumor suppressor, and p63 are often dysregulated in HNSCC. Now my lab focuses on p63. It's a transcription factor that we love, and we study in both a developmental as well as a cancer context. So to give you guys a little bit more background on p63, it's actually a master epithelial transcription factor, which works to maintain the basal proliferative layer of the epithelium. So on the left here, we can see some immunohistochemistry looking at p63 staining. So the p63 is actually this brown stain right here. And we can see that it is highly enriched in this basal bottom proliferative layer of the epithelium. And we know that it's extremely important to actually maintaining epithelial tissues based on our p63 knockout mice. So on the top here, we have our p63 wild type mice, and on the bottom, we have our p63 knockout mice. And if we just look at the gross morphology of these mice, we can see that there are obvious differences between them. Specifically, we see a complete lack of limbs, and there also looks like there's a severe skin phenotype, which when we actually look at histology from epithelial tissues of these mice becomes even more evident. So we can see in our wild type p63 mice, just as you would expect, there's a very nice looking stratified squamous epithelium. However, in the mice lacking p63, we see a complete loss of the stratified squamous epithelium.
Alex, I'm sorry to interrupt you. One of the, Dr. O'Donnell said we can't see the slides. Is that a general? I didn't want you to keep going if you couldn't see your slides. Maybe stop sharing and resharing or something.
Okay, I can see them just fine.
Okay, some people can, some people can't.
I'll stop sharing and go back in.
Okay, thanks. Sorry to interrupt you.
Oh, don't worry about it.
Yeah, I see them now.
Okay.
Perfect. So here we go. So Dr. O'Donnell, I'm glad you can see the beautiful pictures of our p63 knockout mice. So we can see that because of this lack of stratified squamous epithelium, that p63 is extremely important for actually maintaining the signaling and the structure of these tissues. So as a master transcription factor, p63 has been found to actually regulate the expression of a large amount of genes. And specifically in a cancer context, it's been revealed that p63 signaling is involved in pretty much all aspects of tumorigenesis and cancer progression, from angiogenesis to invasion and metastasis to proliferative signaling. p63 targets have been found to affect pretty much every hallmark of cancer. So to actually study and figure out what p63 is doing in HNSCC, we wanted to generate a p63-driven HNSCC gene signature. And the way that we did this was first by creating 2 p63 knockdown cell lines, where we used 2 independent shRNA to actually reduce the expression of p63 in our HNSC cell lines. We then performed RNA sequencing to actually look at transcriptomic changes that occur upon the loss of p63. So just here, I have a selection of differentially expressed genes and some of the pathways that they actually target. And we can see that, as expected of a master transcription factor, there's DEGs that cover pretty much all pathways. And then to also find more direct targets of p63, we've also performed chip sequencing for p63 within our cell lines. So chip sequencing is a technique that will actually allow you to look at the binding of factors across the genome, so you can identify genes that are actually directly targeted by your factor of choice. And from these 2 experiments, we have identified a slew of genes that we're interested in actually studying downstream effects in HNSCC. Today I'm going to show you one of our favorites, MED15. So upon loss of p63, we also see a subsequent loss of MED15 expression. And we also see that MED15 is highly targeted by p63 in HNSCC. So MED15 is actually a part of this mediator complex. So mediator is a multi-protein complex that is a co-regulator of transcription in both yeast and mammalian cells. And it serves as sort of a functional bridge between transcription factors and enhancer sites and the basal transcriptional machinery, including Pol2 and general transcription factors. Mediator complex can be subdivided into 4 different modules: the head, the middle, and the tail on the CDK8 kinase module. The head and the middle module are more of a central core, which are necessary for transcription, while the tail and the CDK8 kinase modules serve more regulatory functions. Now, MED15 is actually a member of this tail module, suggesting that its role is through interactions with transcription factors and enhancer sites. So this leads me to the model that we proposed in our lab, where P63 is driving the expression of MED15. And then there's sort of a 2-pronged model, where first of all, we hope and believe that P63 is actually interacting with MED15 to control the expression of downstream P63 target genes. As we know that MED15 is part of this tail module, which actually interacts with the transcription factors, we do hypothesize that MED15 also has P63 independent responses. So through my research, we hope to both explore a P63 dependent response for MED15 as well as a P63 independent response. But to actually explore what MED15 is doing in HNSCC, we followed a sort of similar approach to P63, where we knocked down MED15 using 2 independent shRNAs in 3 different HNSCC cell lines. We then performed RNA sequencing on SSC25 and SSC29, although for this talk, I'll only be talking about SSC25. And then we utilize our RNA sequencing to actually look at transcriptomic changes that occur upon loss of MED15 expression. So when we actually perform pathway analysis of the differentially expressed genes that we find when we actually lose MED15 expression, it reveals an enrichment of pathways associated with stem cells. There we go. Oh, I forgot I lost my laser pointer. So we see a enrichment of stem cell pathways, immune response pathways, and for the story that I'm going to be talking about today, we also see an enrichment of pathways associated with the epithelial to mesenchymal transition. So I didn't know what EMT was in college, so to give you guys a little bit of background, EMT is the transdifferentiation of polarized epithelial cells into a more mesenchymal phenotype. So basically, epithelial cells will lose their epithelial-associated gene expression, and this will affect the tight junctions and the adherent junctions of these cells and allow them to become more mesenchymal and more motile. And this is important because it has a lot of implications in metastasis, resistance to cell death, as well as resistance to both chemo and immunotherapies. So EMT has been implicated in outcomes in a lot of different cancers. So actually explore what, just because we see an enrichment in our DEGs, like do we actually see an actual EMT signature? We first wanted to look at the expression of some EMT associated factors in our MED15 knockdown cell lines. So just to orient everybody, factors that are in green are associated with an epithelial phenotype, while factors that are in red are associated with mesenchymal phenotype. So when we look at a Western blot of some of these factors, when we first look at factors associated with mesenchymal phenotype, we see that upon loss of MED15, we see an increase in fibronectin and an increase in ETS1, which is a transcription factor that's been associated with mesenchymal phenotype. But we don't really see any changes in fomentin or N-cadherin. When we look at the epithelial factors, we see a loss of SOX15, which is a transcription factor that we have shown is important in the epithelial phenotype. And we see a loss of E-cadherin, which is associated with epithelial cells. So this suggests that there does seem to be a little bit of an EMT program going on, although it may be more of a partial EMT. And this becomes even more evident when we actually look at the RNA sequencing from our MED15 knockdown. So in general, we can see genes that are associated with the epithelial phenotype seem to be decreasing upon loss of MED15, while genes that are associated with mesenchymal phenotype seem to be increasing upon loss of MED15. So again, it does look like, in general, there seems to be an EMT signature associated with loss of MED15 expression. But just because we actually see this gene signature does not mean it's actually having phenotypic consequences on these cells. So just to show you a little bit of some of the phenotypic consequences, we first wanted to actually look and see if the invasive ability of these cells actually change. And we do that through a transwell invasion assay. So what happens is you actually plate your cells in this upper chamber, and in the bottom chamber, you have a chemoattractant to make the cells actually want to move down into your bottom chamber. But in the upper chamber, there is a matrigel layer that acts as a sort of basement membrane that cells have to invade through. So more invasive cells will be more easily able to go through this layer and actually get to the bottom chamber. So after 24 to 48 hours, you can actually stain your cells and quantify the amount of invasive cells. So we perform this at both 24 and 48 hours, but let me first take you through the 24 hours. So just to orient everybody, I have 2 different replicates, and then we have our control cells on the top and our 2 Med15 knockdown cells on the bottom. So we can see in our control cells, there's not really any purple cells showing up, so there doesn't really seem to be a lot of invading cells. And we also see that within our first Med15 knockdown. But when we actually look at our second Med15 knockdown, it does look like there's an increase in invading cells as compared to the control. And this becomes even more evident when we actually look at 48 hours. So again, we can now see that our control is showing invasive cells. But when you actually compare it to the 2 Med15 knockdowns, and especially our second Med15 knockdown, it does seem like these cells are invading more than control. So not only do we see this gene signature associated with EMT, we are also seeing phenotypic consequences that we associate with EMT. So that's just a very small part of the Med15 project that I'm working on right now. But just to give you guys some future directions about where we want to take this, we're going to perform some more phenotypic assays to actually further explore EMT within these cells. We're also utilizing chip sequencing to assess Med15 binding throughout the genome to see if it overlaps with P63 binding and suggests more of a P63 role with Med15. And we've also recently created a Med15 overexpression system as a sort of complementary system to study the role of Med15 in the opposite program to EMT, or the mesenchymal epithelial transition. We'd also like to use it for more phenotypic assays. And the part that I'm most excited about, we're also going to be going into some proteomics to actually find some potential Med15 interactors. But to give you guys a little bit of significance of what's I just presented, the hopefully novel P63 regulation and mediator complex actually influences key genes in HNSCC. Hopefully my mechanistic studies of Med15 will reveal novel insights into both mediator and P63 biology. And we do believe that this will hopefully have implications for therapeutics in transcriptional addiction programs, as we do believe that these interaction between P63 and Med15 could reveal some potentially vulnerable target genes that we could actually target for therapeutics. And with that, I'd like to acknowledge my lab, my mentor, Sat Sinha, and all my lab mates and friends. And up here, I have my contact info. Just like Naomi said, I'm always willing to talk about grad school or science in general. So reach out if you want to talk, and I can take any questions.